All right, everyone. We're going to end on a high note here. Andy and Xavier are co-founders at Data Fellas, so please welcome them right now.
We didn't say anything yet. Right? Go ahead.
So we're going to talk today a bit about analytics on genomics data. This gives a context in genomics, but we're more interested in the process we put in place to get this data analyzed and get streamlining the processing and having services available on top of these analytics. The outline is that we will make a brief introduction to what is genomics data, so you have an idea of the scale of the data sets we want to manipulate and the problems and challenge we want to tackle. And of course, the distributed technologies are here to help, and that's why we talk about in this Spark summit here. But distribution is not only distributing the data. You may have teams which need to collaborate, and they're located in different places. So it's also distributed from different places in the world we want to deal with. And if you think of research process, data is accumulating over a long time period. There's a distribution in time as well to be dealt with, and that's why we need efficient systems for sharing the process. Just a word about us.
Shall I introduce myself?
Yeah.
Okay, so if you hear me, right, so I'm the gorilla, right? I'm Andy Petrella. Yeah, I'm a mathematician. Yeah, okay. I didn't really chosen that. So I did a lot of stuff in geospatial. I did a lot of stuff in distributed computing since my mathematical background, by the way. I created the Spark notebook, if you know it. Yeah, I'm a trainer as well, so I have to leave. And yeah, since I did math and did a lot of stuff, I had to learn. No, actually, I left the machine to do it.
So I'm Xavier. I started with physics because I knew that mathematics per se are not really useful unless you do something with them. And then I moved to biological data processing and using more and more distributed technologies and going to Scala and Spark. And with machine learning as well. Yeah, next. So, yeah, that one we can skip because we already exposed. What is genomic data? So we're talking about, DNA, and as a reminder, DNA is a kind of template that is really a strong determinant of what will be the content of this biochemical soup that we are. And another influence into this reaction is, of course, the environment. But these DNA molecules are really important. And the interesting thing with DNA is that it's inherited. So we get a copy of the DNA from our parents. Every cell that multiplies gets a copy of the DNA with some little changes from time to time that are called mutations. Now, if we think about what's happening on the production side of this data, the biotechnologies have been evolving very fast since the early 70s. And if we compare the amount of production of data to the Moore law, it really looks like this flat, boring line that is the expansion of IT resources compared to the data. And this actually applies to many fields. It's not only genomics. So what is the data? I said DNA, it's a molecule made of small elements that are chained together as a kind of string. And these elements are called nucleic acid, and there are 4 kinds of them. So it means that the data can be represented as a string with letters taken from a 4 letters alphabet. So basically, it's very simple data. And it's 3 billion characters for determining one human genome. So it's not that much. No, there are, of course, other problems. When you want to get the genome from an individual, what happens is that it's a chemical reaction to get that data. You take one chromosome, you multiply it many times, you chop it in random chunks of 100 or 200 base long, and then you get the sequence from all these small chunks with some error at each position, and it's all mixed up, and you have to find out the original sequence. So imagine you take a book, you take sentences or chunks of sentences at random, you make some edits into that, you give it to someone, and you say, OK, give me the book back. So it's quite a difficult problem here. But then it's not only that. Once you have the genome with a certain error on the plate, actually what you want to know is what happens, what is the difference between different individuals, what is the impact of genomes on your susceptibility to disease, reaction to drugs, or things like that. So you need to look at what is variant from one individual to the next. That means that you have to accumulate information on how many, on the different variable positions in this genome. So that's good news because it's kind of reduction. From this 3 million, you get down by a factor of, let's say, 100 or 1,000. But then when you think of the number of samples you get, well, we're 10 billion on Earth, and that's only for humans. So we will accumulate many data sets that cannot be managed in a single data center. It's distributed all over the world. Now we come to Spark. We see that we have a domain where we need some specific representation of the data in form of classes, an API, whatever. And the guys at Berkeley in the AMP lab, mainly Matt Messi and Frank Notaft, developed this Adam library that is really targeting genomics for Spark. And if you look at, genomics data has been accumulating since the 70s and the 80s. And by the time, what happened is that if you want to represent data, you take a committee and they will, in a nice PDF, define how you will structure the file to store the data. But that's not really cool because we work with distributed technologies and these formats are not the right way to go. So the Adam library is mainly stating that the data should be represented in a schema, which really makes a lot of sense. And for this, they use the Avro, of course. So we have a schema. We can generate the classes. We have an API to represent the domain knowledge. And it integrates very well with Parquet storage. So we now have the data that is well-defined in structure. It's distributed correctly on the cluster nodes. And of course, on top of that, they provide specific functions to manipulate the data with Spark. Now, what do we do with this data and these technologies? Well, as I said, we have to assemble these genomes from the raw data that is produced from this sequencer. We don't do that at DataFellas. We're more interested in the analytics part, study the variation between individual and populations. What is the impact of genomics variation on our health and things like that. To do that, we do descriptive statistics, we do machine learning, we do things like unsupervised learning to make population stratification. In marketing, you would call it segmentation of your customers. That's the same idea. Or you do supervised learning, so being able to correlate some clinical data with genomes and with effect on drugs and things like that. And you want to share this process, and you want to be able to replay it, because data is accumulating over time. Biology is not a perfect science. We make assumptions. We have models that are validated to a certain extent, and then new data comes in, and so we need to check that these models are still valid and elaborate more on them. So to do that work, how we do? We actually use the Spark notebook, and Andy mostly develops it because it's the right tool, we think, to work with Spark and this kind of analytics. So every notebook has the same thing. They are self-described and they are consistent. So you take a notebook and it does something every time, okay? And you have annotations, so it's pretty cool to share with others as well. But at this stage, what you do is that you're sharing code, right? You use Git or something, and you're able to share the content of your analytics. So it's mostly say what I do. Okay. Now the Spark Notebook has some feature, is that it's really focusing right now on Scala. So you have a strong help from the type system. You know every time what you're doing. And it helps also providing production quality code out of the box from the data scientist. Because we believe that data science is not only finding that there is value in the data, it's also producing something that helps extracting the value from this data and put it into production. Okay, then it's reactive in terms of charts. So you can have reactive charts with a Scala API, which is cool. It's easily installed, and so you get the package and you simply run it. So you don't need an installation of Spark. You can scale it down to your laptop in a very easy way. And it also manages multiple Spark contexts, which means that you can try something in local and then run another notebook on the cluster in parallel. Okay, so this is a screenshot, a terrible one, I would say. This morning I was looking at me in the mirror and saying, oh, should I show this? It's terrible. It was written a couple of months ago. Why I said I decided to show it anyway because it shows that Spark is really evolving fast. And when you see this, it's a really streamlined way of writing. I want to make statistics on my data and make an histogram. If you would write it now with the data frame API, I think you would squeeze it down to a factor of 2 or 3. So I left it as an exercise for the audience to convert it. But so we edit cells, and we see that we manipulate the types from Adam at this stage.
We gather the copy at the end of the talk, right?
What?
We gather the copies after the talk.
Yeah. You have 18 minutes left to solve the thing. And then we can plot, some histograms. So for example, here we check what is the distribution of mutations that are not very frequent in the populations. And we see that many of them are really rare. So we have mutation occurring all over in our bodies, and they take time to spread in the populations. And then another example, just to show the kind of things that we do. This is population stratification, and we see that with k-means clustering on the data. So we want to separate different genomes based on the data we have on them, and see that the different populations. Here, these are populations going from Britain, one from Africa, and another from China. They separate actually very well. So one of the first things that comes out in the genomic data is the history, where we come from, where our ancestor went through the world. So that's the kind of thing that we can do. And it's not just silly examples. This is really important, segregating your populations before applying models, because you might find some spurious correlation just not considering the heterogeneity of your data set. Okay, so we've seen that we do some stuff with the Spark Notebook, and we're able to share and replay it because it's in notebooks. That's really fine. But we actually want to get much further than that. So the thing is that, how do we share the data we just produced with the notebook? How do we share the source that were used? Because we will replay the notebook, getting data, but maybe it's different this time because the source is changing. And in some way, we also want to be able to publish the notebook as a service. We want to run it again on the data. And this is where I will actually stop, because Andy will elaborate on this right now. So Andy, please.
Okay, I'll take the ground. So yeah, actually we did some older stuff than just k-means, which is not really interesting. We did a lot of stuff with LDA, for instance. We combined k-means with random forest, and we're working on some deep learning algorithms to go a bit further. So right, okay, enough. So now I'm going to discuss more about the different characteristics of a genomic project. It can be applied to other kind of projects, but here I'm going to focus on a projects and research in genomics. And what we think in Datafela is that where we need to go in order to tackle the problem efficiently. So So what is exactly a project in genomics? A project in genomics, interestingly, is including a lot of teams. And those teams are not generally collocated into a single place. They are quite distributed across the world. So you have teams in the US, you have teams in France, in Belgium, in Italy, whatever. And they have to collaborate in some way. So, and not only there are laboratories, but you have also companies, and they have other focus. Not only those teams are distributed, but a lot of people are involved in those teams with different skills. You have biology people, you have geneticists, you have IT guys, a bit like us, medicine, you have some legal, of course. Those guys are also involved in this kind of processes. And not only that, you have also marketers and so on. So a lot of different skills are involved in this kind of project. So, so I talk about the teams, I talk about the people, but also the data are very, very, very heterogeneous. You have biosamples, real blood, right? Piece of skin, right? You have this kind of data where you want to, which you need to process. Imagery, PET scans, for instance. Textual, actually genomics are textual data, ACTG, and they are, sadly enough, distributed in this way. But archives are very, very important. They may be the most important data that you have to consume, not only genetics, but a lot of different kind of data archived in historical data, yeah. So another characteristic of a genetic or medical or health-oriented project is that you have to gather many people, not only teams, but also people like us, right? Actually, we are the testers for those guys. So we need to be gathered at some place several times across several years, maybe, to be checked, measured, and so on, right? We're not really testers, but also because we are trying new medicines and we are sick, and we get some added value by participating into that. But still, they have to gather you, they have to contact you, and it takes a lot of time. And of course, it takes a lot of money, because it's not always free. Some examples of projects in genomics. For instance, the 1,000 genomes that has started in 2008 and ended 3 years ago. So it's only 1,000 genomes that they have been collected. We are a bit more than 7 billion on Earth. So it's a very small fraction that has been collected at some point in time in 4 years, involving many, many teams across the world. The world. And it generated like 200 terabytes of data, which is, okay, not that much. But another project that started here in Britain is the 100,000 genomes project, 4 years, again. So we scaled by 10 in 4 years, 5 years. 20 petabytes of data, most probably more, because we are collecting more and more data. And in the US, you have the 1 million genomes project that has started. And again, 4 years, right? So it's always 4 years, right? But the factor of data that we are adding is 10, right? So we are at 0.2 exabyte of information. However, this is just genetics information. This is just a sequence of information, right? You don't have much information than just this. And we only collected like a small fraction, 1 million over 7 billion. There is another kind of project, EQTL, for instance, which is trying to match part of the genes to characteristic on the body or reaction to some medicine and so on, which is then involving a lot more data, right? So you have this exabyte of data, and then you have to multiply it by 100, thousands, you don't know, right? Now, this is the kind of project that you have, small projects, by the way, in 4 years, just 4 years. So that means that you have many teams, many guys, many kind of data, so you need to really have a proper data management systems to do that. By still coping with the fact that you have a lot of data, heterogeneity, distance between actors, and constraints. Yeah, we have laws, of course. I mean, between France and US, you cannot send whatever. It's painful for the science, of course, but it's like that. You have to cope with this fact, so you cannot send everything. So you have to expose services that are stripping out some data out. Right. Thanks. So what we think is that one of the ways, maybe the only way to do that, is to go towards a system that is distributed friendly, for sure, but also is integrating the notion of schema and binary, not having ACTG in a text file, but really binary information like Avro and the Parquet behind. So Avro is a schema, then Parquet can be the binary storage. Right, research now. So it was about the project, not research, mainly research, right? In medicine and health, this kind of projects are very, very long. So long. So for us, maybe in IT, we think a project of 4 years is long. There, those guys are just kidding at us, right? They're just laughing at us, right? So they're very, very long. And one of the factors why a project in medicine are long, you can go to the next one, they are more or less obvious, right? But still, they mustn't be overlooked. You have a lot of measure to do, a lot of validation to do. We are talking about health, right? People, life, right? You cannot overlook those kind of things. There is a lot of control, so when you have a result, you need to send this information to government. They have to check what you've done, right? It's just like, okay, this is the service. Yeah, we have a good SLA, right? No, it's not like that. They have to check the sources, they have to check the processes and the results, right? And of course, you have a lot of actors. The testers, the medicines, the validators, everybody is part of that game. So not only the project by itself is tough and very hard to manage, but also afterwards you still need to apply a lot of layers on top of it in order to be able to distribute your product. So, right, so a matter of fact is that actually research is a function. It's a function that goes from data to results, right? We can resume it to this kind of fact, right? But what happens if we lose the data? Historical data, whatever data. What happens if we lose the results? We lose money, we lose time, we lose lives, right? This is already painful. But we can get into troubles very, very, very soon by just simply forgetting about storing the processes, forgetting about how the data has been processed, how the results has been produced. This information is very valuable and needs to be also kept and shareable as much as possible, maybe even more than the data and the results themselves. So why? Because actually when you are producing some results, you need to have a scientific behavior. That means that, okay, I want to confront that with other systems, with other models, with other data sources, with other results, right? You want to replay it, right? Otherwise, you cannot really assess that. And finally, being able to enhance a process is the way science is evolving. Always reinventing the wheel, always reinventing new models is not always good, right? So you still try to move towards the right direction by taking something that has been done and trying to squeeze it in another direction. So, yeah, it would be misleading to think that just sharing the code then is enough, right? The code is not enough. It's good to have, but it's not enough. For sure, when we do research, we mainly we look for data, results, not really for code, right? That means that a research process is including code. Okay, that's fair, but the context as well. How this code has been generated, why it has been created, when, where, right? This is the context of the research that has to be part of the system, right, and the process. The sources of information, of course, you need the data in order to be able to replay it and to understand what the guy wanted to do. Otherwise, you have a result. You are talking about life, right? You want to create a model. Do you really want to use data that you don't really want, that you don't really know what is the accuracy? You don't know if there were some measure problems at the start of the processes. You don't know anything. And you say, okay, no, it's good. No, take it easy. We have a good confidence interval, right? So go ahead. But you don't know the sources, just dump. So one example of research in health can be like, okay, we want to assess the risk factor associated to a disease given a mutation. Because the mutation is quite easy to have. We just sequence the guy, we have the mutations, we compare to the reference, we know the mutation. So yeah, why? What could he have in like 10 years, 15 years? This kind of project takes like 50 years to run. 50 years collecting data, modeling, hundreds of researchers, different generation, new models, right? What if you're not able to rerun the processes that you've just created on the old data? Because you just don't know what they are. You just forget about them. And the reverse is also interesting. What if you're not able to run old processes onto the new data to be able to assess with things that you already used? These are all about replaying, but across time. So all these facts lead us to think about something really shareable, right? We want to share as much as possible. So a good project will be able to go to until the target, to reach the end efficiently if they are able to share as much as possible information across teams, people, and so on. So we want to be able to share data efficiently, accurately, but also the results, which are in some sense data as well, and the processes, of course. This is the idea of share. The challenge, by the way, resides in the workflow. How the heck will I do that? Okay, you want to share, but now how? How can I gather enough information? How can I share it? Where do I have to put that? How do I communicate, right? You have so many skills how to do that. So a workflow today in distributed computing involves a lot of stuff and a lot of people, right? So you start with creating a cluster. You need the ops. You go to the sources. The data engineer is responsible for that. He finds the resources maybe, create the context around it, the quality, the semantics. Then another guy came in the game and he connects to the sources. He says, OK, the Cassandra is there, right? And the data is the schema. Then you go to creating a data model, a distributed data model. And that means that the scientist has to be part of that game. You need to tune the accuracy again, looping with the tuning the performances, which involves so the ops in this part. Then okay, you have a model, right? So you want to really provide to the customers or the patients just a histogram bar table? You don't want to do that because at that time it's just a snapshot of your results of your data, right? What you want to do is write this information back to a sink. Because the data you just produced, your model, your prediction, is also a new data set that you want to reproduce, to replay or to reuse in other fashion. I mean, you need to write to sinks. So also the ops and the data engineer are part of that game. Then, okay, so you have Cassandra, you have Parquet files, but what? Who's gonna consume this information, right? How could we consume this information in a scalable manner, right? So for that, you need web services on that, right? For web services, you need to contact the web engineer and so on and so forth. And finally, when you have these web services, you need to render data charts, data dashboards, new web services connected to them. You have plenty of different consumers, but just another layer. Just don't publish tables. This is not a data, right? This is just a snapshot of the world. So we talked initially that we are using a lot of different tools for that in our stack. Actually, yeah, for sure. Not only those guys, by the way. But if we need to create a cluster, OK, fine, Mesos is there. Spark for the cluster, for the computation. Tachyon for some in-memory cache. And OK, so I'm not going to list them because I just have 2 minutes. But by the way, we are losing Tachyon a lot, as you can see there, because Tachyon can be used in different manner, like to cache intermediate results or just pre-computed views in a time and space manner. So, gonna finish stream. So the idea here is that you have too many people even in the process. Too many people. So they're doing a Python, Scala, Perl. Hi, guy. So maybe sometimes some Fortran, and then they can really talk to each other. That means that we want to streamline these things, right? So we want to be able to share as much as possible information without having to redo everything from scratch. So that's why we have this notion of share, this kind of toolkit that includes many tools like Mesos, Spark, and Tachyon, but also Cassandra, Solar, Elasticsearch, and of course Mesos. So we go from the notebook, we generate project, we generate some web services in Avro to keep the schema. Then from there, we can connect Tableau, ClickView, dashboards, your own dashboards. But by the way, We have notebooks, we have web services, we have Avro schemas, everything can be indexed, right? Even the model itself, even saying, okay, actually this guy is processing information to do some disease discovery, and he used this model, and this is good information to keep in mind, right? This is information that need to be discovered at some point in time. So you put everything into a database, and you have a semantic context around your system. That means that one guy can come into the Span notebook and say, okay, I'm gonna look for certification or to EQTL processes, and you can be presenting so many services, sources, results, notebooks, information, models, whatever, in order to bootstrap your work. And this is sharing knowledge. Right, and if you want to know more about what we do, so you can still contact us. We are always on Twitter. We have a website, of course, but also, yeah, we wrote a blog post about what we just said at TypeSafe, because TypeSafe is very interested in the notebook, so we've been invited to do that. And by the way, O'Reilly has been interested in our work. So that means that in March next year, we're gonna run an online course, right, online training, where we're gonna demonstrate how we can chain those tools all together, from Mesos, Spark, Kafka, Cassandra, how to tie them up correctly. It's a short training, but interesting enough, in my opinion, to be part of. Of course, you cannot click on that, right? So we're gonna publish it on Twitter soon. Right, and I think it ends our talk.
I have a question regarding the data that you have here, that you have the numbers about the data that you have shown. I would expect the genome data to be exceptionally to compress exceptionally well. I would perhaps expect 100 times compression or even more than that, because it's so similar. So the numbers that you showed, were they raw data or actually compressed?
The 200 terabytes?
Sorry?
The 200 terabytes?
Yeah. Or
This is the data which are generated. So this is the data that are currently generated. So it's the raw data.
The raw data, okay.
The compressed data is around 40 terabytes, something. There is a factor of 5 at least, up to 20, depending on the data that you're compressing.
Okay, yeah, thanks.
All right, let's give him another big round of applause.